All right, so thank you guys for joining us today and attending our workshop. My name is Lay.
Oh, my name is Casper.
And we're both with Freshwater Conservation Canada. We're really glad to have this opportunity to share some of the really cool work that we've done over the last 7 years, and we appreciate everyone taking time out of their day to join us to celebrate some of the successes that we've had with the Native Trout Collaborative. Before we begin, I just have some housekeeping items. Bathrooms are back there, but you can access it through the hallway through the back, so we're not going in front of the screen. In the case of an emergency, please exit through the front doors, and the muster point is in the parking lot. Also, there's a cord, as you guys can see, there's a cord that goes across, so please be careful if you're going across it.
Excellent. So today we're meeting on Treaty 7 land. Bridgeland is located just north of the confluence point between the Bow and the Elbow River. The Blackfoot name for Calgary is Mokinstis, which is in reference to this point. This land is the traditional territory of the Blackfoot Confederacy, which includes the Siksika, the Piikani, and the Kainai First Nations. It's also the traditional territory of the Stoney Nakoda, which includes the Chiniki, the Bearspaw, and the Wesley First Nations. This land is also the traditional territory of the Tsuut'ina Nation and is home to the Métis Nation of Alberta, Region 3. So Leigh and I are just going to do a quick intro for the collaborative before we get into our main presentations.
We're also advertising a really fun trivia night and social event that we're having tonight. It's at 5:30. And it's at Courtyard, which is just on Edmonton Trail. It's about a 4-minute drive from here. Some of you guys might already be signed up for that event, but if you're interested, please let one of us FCC staff know, and we can get you some tickets. And we have really cool prizes donated by Patagonia. So a little bit about the Native Trout Collaborative. We were formed in 2019, and we consist of a group of organizations working to advance native trout recovery in Alberta. Some of our members include Freshwater Conservation Canada, Canadian Parks and Wilderness Society, or CPAWS, the Southern Alberta chapter, Cows and Fish, the Government of Alberta, specifically the Environment and Protected Areas sector, FRI Research, and Alberta Conservation Association. But we've also closely worked with various organizations like the Bow Habitat Station as well. So the goals of the collaborative were to coordinate resources to mitigate threats and promote the recovery of Alberta's threatened native trout through things like habitat restoration, recovery stocking, land use planning, and watershed and fish population assessments. But we've also worked really closely to help educate, engage, and inspire all Albertans to take pride in our native trout and to take action to help care for these habitats. We received funding through the Canada Nature Fund for Aquatic Species at Risk from the Fisheries and Oceans Canada. It has funded many of our projects and outreach initiatives, as you can see from our many photos above. Unfortunately, as of March 2026, this funding is ending, and the Native Trout Collaborative will be disbanding. Regardless of this, all NTC members will continue to commit to protecting our native trout in Alberta, and we hope that everyone here will also join us in this mission and continue to support our members. As most of you guys know already, in Alberta, we have 3 species of risk, 3 species of native trout that are at risk. These include the bull trout, West Slope cutthroat trout, and Athabasca rainbow trout. All these species are listed as threatened under both the Alberta Wildlife Act and the Canada Species at Risk Act, with the exception of the Athabasca rainbow trout, which is listed as endangered in Canada.
All right, So we're going to take a quick look at these maps. So this is probably, for some of you, not the first time you've seen these maps. So these are Fish Sustainability Index maps. They were developed by the Government of Alberta in 2018 and 2019, and they compare the historical and current density of our native trout species in Alberta. So the first one we have up here is the bull trout. I think I said it, but the historical map is on the left and the current map is on the right, or current as of 2019. So these maps are a little bit out of date. But the main thing that we want to take from this is that on the left, on the historical side, we have a lot of green. So these were very high and high density populations. And now the colors kind of change up. So we have a lot more red, orange, and yellow, which is moderate, low, or very low. And we've got more areas that are considered to be functionally extirpated. Move to the next one. This is the West Slope cutthroat trout map. As you can see from the map on the right, we have a lot of areas that are functionally extirpated. I'm sure most of us are familiar with the term, but these are areas where the populations are considered to be gone, or if there are individuals there, there isn't enough for it to be a sustainable population. So for the West Slope cutthroat trout, you can see that their populations are being pushed to the west. So all of their populations are just like on that band near the Rocky Mountains, and we've only got some red and some orange, which is very low and low abundance or densities. And last but not least, we have the Athabasca rainbow trout. So the Athabasca rainbow trout is only found in the Athabasca Basin, with other rainbow trout populations in Alberta being introduced in the past. So as of these maps released, there was no areas considered to be functionally extirpated, but as is the case with all three of the maps, there are likely populations that have already blinked out that are no longer considered to be there. So as we can see, we had lots of green, and now again we've got very low and low populations in their region. So there isn't a single reason for the decline in our native trout populations, so we chalk it up to cumulative effects. So past and present impacts have contributed to the decline of our native trout. So some of these impacts you're probably very familiar with. So we've got habitat loss, changes in water temperature and quality, and of course issues such as climate change are accelerating these issues and continued development in their critical habitat. One big issue that I think we'll hear about more today is hybridization. So hybridization occurs when two different species reproduce and produce a hybridized offspring. So this is a particular concern for our native trout species. So West Slope cutthroat trout and Athabasca rainbow trout reproduce or hybridize with non-native rainbow trout species, which is a big concern. So hybridization dilutes the beneficial genes that our native trout have developed over thousands of years, and a decrease in pure native trout in whichever species also has impacts for their ability to be designated under federal and provincial acts. So they aren't protected anymore if they fall under 95% pure. Additionally, bull trout can hybridize with non-native brook trout, which is also a concern. So that's our quick and simple intro to the Native Trout Collaborative. So this is our schedule for the day. We're going to have 6 presentations. So we're going to start off with 3 presentations. Each of them will be followed by a 5-minute question period, and we'll try to get to as many questions as possible, but just because of time constraints, we might not be able to get to everyone. After our first 3 presenters, we'll have a 10-minute break, and then you can go to use the washroom, grab some snacks, and then we'll come back together for our last 3 presentations. And then once we've completed all of our presentations, we will be doing the draw for our door prize. So if you haven't entered the door prize and you want to, make sure you do that during the break. We'll be getting into that right after our presentations. So without any further delay, we'll go right into the presentations. So I'm going to, first up, we have Mike Roedke, who is presenting online. So I'm going to get that set up, and hopefully I have no issues. Okay. Okay, Mike, I think you can share your screen now.
Okay, and you can hear me okay?
Oh, not yet. I think our Bluetooth speaker disconnected. Hold tight.
Okay.
Oh,
Oh my god, I felt that
okay.
was coming from the computer.
All right, we can see your presentation.
Okay.
Is there a way to full screen?
No, I think he just hasn't full screen.
Yeah, it's fine.
Okay, but sound.
Yeah. Oh, sorry. All right.
How are we doing?
We're almost there.
Okay. And I haven't gone to full screen yet, so.
Okay, it's there. It's coming back. Okay. Go.
It's connected.
Power on.
Oh,
Bluetooth mode.
there we go.
Bluetooth connected.
Great.
Excellent. Okay, give that a try, Mike.
Okay, how's that? We good?
Do you want to speak through that
Still-
one instead?
Mike, can you speak
Still
again? just so we
connecting?
can see if it's
Sure.
working?
Testing, testing, 1, 2, 3.
Is it coming out? Oh. Sure. Try that. Okay. I like can't see both. Okay. Where's is the mouse there? Okay.
There you
Go to Mike.
go.
And then change the mic to the speaker to your. It is on the. Crank that volume possible.
Okay. Mike, can you hear us now?
I can, yes. Can you hear me?
You might be able to hear him out of this one.
Oh, it's connected to that computer instead?
Oh, like headphones.
Oh. Mike, can you say something now?
Sure. Is it working? Oh,
It is definitely working.
it just deafened everybody.
Mike, you can start whenever you're ready.
Okay, we're good now?
Yes, you're good.
Okay, very good. Yeah, and I guess just jump in if something goes wrong here. Thank you very much, everybody, and sorry I couldn't make it there today. Yeah, I just wasn't in the cards for me today, but yeah, I really appreciate making this happen. So I'm going to talk to you today about working smarter, not harder, an evaluation of Alberta's 300-meter backpack electrofishing standard reach. So a little bit of background, for those of you who have worked in fisheries in Alberta, this is probably a familiar document to you. That's the province's standard for sampling of small streams in the province. Created in 2013, but essentially it was formalizing what I would say was an informal standard that we'd all been using since at least the late 1990s, if not a little bit earlier. And the application of this standard is for stream surveys where the principal objectives are fish presence and distribution, community composition, and relative abundance. So the basics of a fisheries inventory. And just to point out that this was a minimum standard. And then getting to the heart of the protocol that I'll be speaking to today, it's the distance electrofished. And specifically, there is the requirement that you do 40 times mean wetted width or 300 meters in a stream, whichever is greater. So we can look at comparable work from elsewhere to see what kind of standard reaches they recommend for similar objectives. And a couple of things I'd like you to take away from this is, number one, there's not a lot of comparables. In particular, there's not a lot of studies that have been done in mountainous areas like the eastern slopes. And then also when you start looking at comparing the conditions we're working under versus some of these studies, a couple of things jump out. Number one is that we're not encountering a lot of species out there when we're doing our work, quite low actually. On average, for our work, it's 2 species with a range of between 1 and 7. And then also, although the 40 times is fairly consistent across the board, or variations on that, the 300 meter minimum is a relatively long minimum reach length. So the objectives for our work were really, we just want to answer the question, considering that our reach is relatively long, are stream reaches that are less than 300 meters adequate to represent fish species richness, presence of native trout, and relative abundance of native trout in east slope streams? So why would we care? Well, we've got the Athabasca rainbow, bull trout, and west slope cutthroat trout are all species at risk in Alberta. They also happen to be popular sport species. And they are typically sampled using backpack electrofishing gear. And hopefully it goes without saying, but nevertheless, I'll say it, precise, accurate, cost-effective data are critical to the management of these species. And this was particularly important for ACA because we do dozens and dozens of backpack electrofishing surveys each summer. Briefly going over methods here. So we picked 221 of the randomly selected backpack sites that we'd worked on over the years. Some of these sites go way back to 2014 or as recently as I think 2022 or 2023. And up and down the eastern slopes, we wanted to try and cover the range of our native trout in the mountains. On average, our effort was about 1,200 seconds, although that ranged considerably. Crew size was 3, although sometimes we had 2 and as many as 5. Our sample time, we measured that as well when we were at the site, and that was on average 80 minutes, but again, ranged fairly widely. One thing I want to make note of is that these are relatively high elevation sites, particularly when we compare them to other studies like ours. And cold water, 10 degrees Celsius on average at the sites that we included in our study. So what did we do while we were out there? Well, we did the standard. So again, if you've done backpack electrofishing sites and streams, this will probably look pretty familiar to you. 300 meter reach, 50 meter transects. At each transect, we were counting our catch, measuring our effort, taking a wetted width, and a maximum depth along the transect. Between transects, we were visually assessing percent pool, riffle, run habitat. At the start of our reach, we would randomly assign duties, and we would also take a start time. And at the end of the reach, we would take the end time. We measured water temperature and conductivity. And a little bit unique to this work is we also measured the distance to first capture of each fish species we detected in the reach. And also just to point out, we started all our reaches at the top of a riffle habitat just to standardize the habitat that we would be starting out in. When we collected all these data, and as I said, we've been doing some of this since 2014, so it's fairly long span. We modeled them using a generalized linear mixed effects model. It was a hierarchical approach, and we placed a prioritization on parsimony. So we were assessing the models using AIC. Predictor variables we included in our analyses included conductivity, water temperature, mean maximum depth, which was a categorical variable, mean wetted width, again categorical, percent pool riffle run. And then we also, in the modeling framework, we accounted for I guess what you could call the nuisance variables of site effects, watershed effects, year effects, and effort effects. Getting into our results here, what we found with species richness first is that we found mean wetted width had a significant impact on the distance of reach that we needed to electrofish to feel confident we detected 90% of the species in the reach. So what that looked like is that top line there, the blue line, that was for streams with a mean wetted width less than 2.5 meters. We were quite confident that we were getting 90% of the species encountered in the reach in the first 100 meters sampled. For wider streams, so between 2.5 and 5 meters mean wetted width, that bumped out to 200 meters. And coincidentally, or perhaps not, both of those work out to 40 times mean wetted width. And then we didn't have a lot, but we did have a few wider streams. And we actually found there for the 5+ meter mean wetted width streams that you needed to go 250 meters to feel confident you detected 90% of the species in the reach. So conclusion we drew from that is number one, well, the 40 times mean wetted width is probably a pretty good rule of thumb. Not only have we found it for our study, but it seems to be pretty consistent across several studies. For the streams we're working in, 300 meters is typically much longer than what the 40 times mean wetted width would require. Actually, for about 90% of our streams, 300 meters was oversampling, if you want to look at it that way. And it probably part of this is because our crews typically did not sample the widest streams. Usually it was felt like it was impractical or unsafe to do so. Impractical in the sense that at a certain point, if your stream is wide enough, you actually end up doing almost more back and forth than moving upstream in the stream. We also found that the strict interpretation of the multiple of mean wetted width to determine a sample reach is actually quite awkward to implement in the field and also hard to plan around. And if you were to make a blanket suggestion, probably 200 meters for a standard reach for species richness is adequate in most eastern slope streams. Moving on to trout detection. And we actually included brook trout in our analyses just to act not as a true control, but as a control, I guess, pseudo control. We just wanted to compare against a non-native species that we're not really worried about them declining in abundance and distribution right now. And what we found was overall that if we were going to detect them in the reach, it was typically within the first 100 meters. Now, obviously, there were some places where that wasn't the case, but overwhelmingly, it was within the first 100 to 150 meters. So when we modeled that, we essentially got two curves. And that was reflecting that we found that the detection probability for these species decreased with mean maximum depth within the reach. So when in the relatively shallow reaches, sample reaches, so under 0.5 meters mean maximum depth, we were detecting trout within the reach in the first 100 meters. So that's, if you take our average mean wetted width, which was 4.4 meters, that's 23 times mean wetted width. For the deeper reaches, up to actually 1 meter, which if you've ever waded out there, that's quite deep. That bumps out to 150 meters or 34 times mean wetted width. And we actually had a few sites that had their maximum depth was greater than a meter, but it was very few. So really we couldn't tell much from that data set. So to conclude, we found that species richness out there essentially meant trout species richness in many of the streams we sampled, because we were only encountering salmonids. Detection probability was greater than 90% in the first 150 meters at almost all the sites. And these results are quite consistent with comparable studies. Finally, relative abundance. So we're looking here at, these are the cumulative CUEs, so that's fish per meter as you increase your reach length at all the sites we sampled for the 4 species. And overall, you'll see that CUE is fairly low and highly variable. And just for comparison, we can look at what's considered high abundance for a cutthroat trout stream in the US for their planning, which we had very few sites where we had that. And actually low abundance, most of our sites would be considered low abundance compared to the States. So how did variability change with an increasing reach length? So we looked at that by looking at how coefficient of variation, which is your standard deviation, divided by your mean, varies with increasing reach length. So these are bootstrap values. And the take home here is that they're highly variable and they really only improve in the first 100 to 150 meters. So why is this? Well, we're calling it the rule of 10. What we mean by that is that when you're sampling a species or making counts of an animal like fish in a stream, and your mean is around that 1 fish per sample site, if you happen to catch an extra fish, well, that's 100% change in your catch. But if you're working around the 10 fish per site and you catch an extra fish, well, that's only a 10% change. And you can see where I'm going with this. You get out to the 100 fish, and yeah, 1 extra fish, give or take, is really a 1% change. So what we were finding is that almost all our sites were operating right in around this mean. So 1 fish made a big difference in our catch. But for a small subset of our sites, actually, as you increased your reach length, you got into this sweet spot of the minimum of 10 fish per site. So then we were seeing that reduction in the variation in our catch. But we almost never caught 100 trout at a site, so we never really saw increased improvement beyond 150 meters. So from this, we're concluding, while our catches were low overall and highly variable, increasing reach length did essentially nothing to improve variation in CUE past 150 meters. And actually only 4% of our sites had counts that were equal to or above 10 trout, which is kind of the sweet spot where you start seeing that reduction in variation. So what this meant was that many of our sites were default presence-absence surveys. And interestingly, we found similar results for brook trout as native trout. So this wasn't just a native trout reflecting potentially low abundance of native trout at our sites. So what does this mean for monitoring of trout in the province? And I was glad to see these maps being used in the introductory. It means I don't have to explain them, but the point here is that we're monitoring these species based on fish density. And I'd like to say this is a champagne tastes, but we have a beer budget. So what do I mean by that? Well, by champagne tastes, I mean, and here's an example from Andy Paul's work back in, 2000, I guess, or something like that now on Eunice Creek. But the point here is these native trout can vary hugely in abundance over a short time frame. In this example from Eunice Creek, it was 2 orders of magnitude over 15 years. So that's a lot of variation in abundance. So how do we deal with this roller coaster of fish abundance? Well, there's a few things we can do. We could try and reduce our observation error and control for covariates of abundance, and we do this. We could also accept higher type 1 error rates, so your false positives. This one's a bit risky, but we could accept a larger minimum detectable change. So we could say, look, we just know we're not going to get the small changes. We'll wait for the big changes. The reason why I think it's risky for native trout is because some of these numbers are so low. If that change is a small change down, that could mean loss of the fish. And then finally, and this is fundamental, is we could increase the number of sample points. So when you're talking about time series, that's monitor for more years. That brings me to the beer budget. So here's a good example of work we did in the Upper McLeod. It was over 2 years, 76 sites sampled, so that's 23 kilometers of stream electrofished. We had 32 sites with 0 catch and 14 sites where we only caught 1 fish of a native trout. So essentially 60% of our sites were effectively presence-absence surveys. And only 9 sites did we catch greater than or equal to 10 fish. So that was that sweet spot, the rule of 10. And the beer budget, it's expensive to do this work. For this specific study, it worked out to about $2,600 per site. Now, I know this is very crude back of the napkin, but my point being is there's 88 Huck 8s in the bull trout recovery area. If we were going to do a single pass at this cost, we're looking at about $17 million. So what the heck do we do about that? Well, maybe there's an alternative, and perhaps that's occupancy modeling. We know it's more robust when counts are highly variable, like we encounter, and we also know it works well with low density or rare species. Sounds a lot like native trout in the eastern slopes. Lower sampling effort requirements, and it can align well with management questions about distribution and persistence. And at one time, you could argue that it's a fairly new approach and we don't know a lot about it, but that time is long since passed. It's well established in the literature. But there are trade-offs, like there always are. Here's an example from a site we sampled in the Ram River. We did it for 5 years. You can see if we just looked at our bull trout catches, there's some really interesting variation going on in there, and it's actually strong year classes. So you had a 6x in variation over the 5 years, and we had to sample 1.5 kilometers to document that. If we were just looking strictly at bull trout presence, well, there was no variation. We caught bull trout every year, and we only had to sample 111 meters to do that. But obviously, you don't get that information about the strong year classes. So there's trade-offs. So what the heck could we do? Well, this may be the one time in native trout management where I'm going to say hybrids might be good. So what do I mean by that? Well, we did a number of sites in the Ram watershed for this work. And yeah, there's our R10. Maybe that's a good site to do the abundance monitoring. We know we're going to get decent catches, over 10 usually in a year, so that it's more likely we're going to be able to detect a change. But for these other sites where our catch of, in this case, it was bull trout, was quite low, 1, 2, 3 maybe on any given year, maybe those are more suitable for some sort of occupancy monitoring approach, where you can really do low effort at a site and do more sites. And then finally, sites like this where we just never ever encountered a fish, maybe those ones should be removed from the sample frame entirely. To summarize, the 300-meter reach is really the multi-tool of sampling protocols, and it's probably best suited to exploratory studies and is relatively inefficient for targeted questions. And I think what we need in Alberta is we need a toolkit for native trout management. And that could look like more effort for our abundance questions, and maybe accepting that we'll just get occupancy at some of our other sites and have correspondingly less effort. So if we do it right, we could get the right tool and maintain or improve our power for equal or less money. With that, I'd just like to acknowledge all the people, well, not all the people, but the people most recently have been working on this with us. So that's my co-workers here at ACA, Jason Blackburn, Kevin Fitzsimmons, and Peter Aku. And then shout out to Nilo, who I think might even be in the audience out there with me, Stacky. She's the statistical wizard behind our analysis. And then critical support, field support, data management, that'd be Brad Herkett, Chad Judd, Emily Franks, and Zach Spence. And the many, many field staff and funding partners we've had through the years. And of course, the Native Trout Collaborative as well. Thank you.
Excellent. Thank you, Mike. I hope you heard the applause there.
I did. Thank you.
Amazing. So I think we've got time for, I'd say, 2 questions. So I didn't see anything in the chat, but do we have any questions in the audience? Sure, Noreen, and then yeah.
I don't know if Mike can hear, but just, I guess, given that there's not enough money or time to do all the sampling, where does eDNA sampling fit in here to get more places?
Okay. So Mike, the question was, how does eDNA fit into this?
That's a great question. Yeah, and I would say that we definitely need to be considering that as part of our toolkit, especially if we're comfortable going for occupancy as opposed to relative abundance.
And just to add to with native Athabasca rainbow trout, the DNA between Athabasca rainbows and non-native rainbows is pretty indistinguishable, I guess, at this point. So there's more.
Okay. Any other questions?
I just have a small one related to, in this case, with Westlaw Native Trout. There's going to be like the rate of hybrids with rainbow trout. So how is it possible to identify that, and do you count them as numbers at least when you're doing the sampling, or what's going to be the process with the hybrids in this case? I don't know.
Okay.
Yeah.
How will the sampling process work with hybridized individuals between West Slope cutthroat trout and rainbow trout? Is that okay?
I guess currently or in the future?
I guess for now, what's going to be the approach?
Yeah, currently and the future as well.
Okay, because yeah, that's an ongoing challenge, and you know there is no substitute for actual genetic determination of the amount of hybridization. We do have field characteristics we can use, morphological, particularly for brook trout, bull trout, much less reliable, obviously, for cutthroat trout. But that is, yeah, just an ongoing issue with almost any approach that we've taken.
Thank you.
Awesome. Okay, let's give him another round.
Thank you.
Awesome. Thank you so much, Mike. Next up, we are going to have Jonathan Gosling from Freshwater Conservation Canada. John, you can come up and I'll try to get your presentation up. Okay, there it is.
No. I think everyone can hear me.
Yeah.
Power off.
Peace.
You can use the clicker too.
Does that thing work to change the slides?
Yeah.
Oh, yeah, it's working. I can walk around.
Sorry, I forgot.
Huh? Oh, so that they can hear me.
Yeah.
Okay. Can't walk. It's not a mic, too.
Yeah, I know.
And do we have to share our screen to Teams so that people online can see?
Yeah,
Huh?
Okay. Sorry.
Okay, good. All right. Welcome, everybody, to my presentation about the Calm Creek watershed in the Porcupine Hills in southern Alberta. My name is Jonathan, as you can tell, and I work at Freshwater Conservation Canada. Yeah, so just, okay, maybe it doesn't work. Yeah, I don't think so. Okay, there we go. yeah, just to give you guys a bit of an overview of the project as a whole that I'm going to be talking about today. So I'll give you an watershed overview, talking about just the Callum Creek watershed, its issues, who and what lives there, our goals in this watershed for the project, and how we aim to increase the health of the watershed through monitoring and detecting change through our rehabilitation work. So here is the Callum Creek Tuck-10 watershed. This watershed is pretty special. It's just about 20,000 hectares in size, but it's special because it is disconnected from the upper Old Man River by a waterfall barrier, and it still contains pure strains of West Slope cutthroat trout. Critically, in two creeks that are tributary to Callum Creek called Plale and Sharples Creek. They have been observed throughout the watershed and other small tributaries as well as the main stem of Callum Creek itself. However, the only two areas designated as critical habitat are those two tributaries. I guess another cool fact is that there's also no brook trout here and no rainbow trout, as I already said. So it's one of the last remaining watersheds that does not have hybridized fish or invasive fish. Okay, so this project aims to address some of the issues in the watershed in a multifaceted approach. So through these 5 pillars you see here, we aim to overall improve the health of the watershed. So this watershed has a lot of grazing and forestry activity, as well as recreation. So these are all things that are causing degradation to the watershed year over year and has been ongoing for a long time. So to address these issues, we've been working with a ton of the project partners in this room. Honestly, most people are here, to study and rehabilitate the watershed in a way where we can baseline condition it and then assess how our rehabilitation is changing and affecting the watershed in what we believe to be a positive way. So our project goals are 5 goals, and they're not really in order here. This is just kind of random. So goal number 1 was to establish the baseline data in the watershed. This includes all those things that we've been monitoring that I'll talk a little bit more to. And I give it an upside down check mark because, yeah, we have 2 years of really good baseline data and 1 year of fish data, but it's something that we're going to keep revisiting and keep building upon for the next couple seasons, so that we can have really good controlled areas for data as well as areas that are being impacted by our rehabilitation. So that's kind of goals 2, 3, and 5 tie back to that baseline data. So we're going to use that to assess how the watershed has changed based on the work that we're doing here. Goal number 4, I'll talk a little bit more about that, but obtain a pre-rehabilitation population estimate of the West Slope cutthroat in the 2 critical creeks that I mentioned earlier. And we achieved that in September of last year. So the monitoring for this work, as I said, began 2 years ago in spring of 2024. So to meet the goals that I just laid out in the project, we needed to get those baseline conditions. So that included temperature, stream flow, aerial baseline conditions, as well as visual monitoring through us and some of our partners. And then lastly, the fish monitoring side of things. So we currently have 24 stations for stream temperature throughout the watershed, 4 stations for stream flows, and have 4 pit arrays throughout the watershed to detect tagged fish moving in and out of different tributaries. And another small portion of the project, which is just starting but is going to become a lot bigger, is the aerial monitoring. So I'll talk a little bit more about that coming up too. So why do we need to monitor the temperature, you might ask? Cutthroat are a little bit picky when it comes to where they want to live. And so they have basically an optimal temperature range that's between 9 and 15 degrees in stream. And they can start to experience stress when it reaches above 15 degrees. And when it reaches around 20 degrees, this is their UILT, which is the upper incipient lethal temperature. It just basically means that when a population is experiencing this temperature, generally speaking, half of that fish population will perish and won't survive any further. So it's really important for us to monitor temperatures throughout the watershed so that we can have a targeted approach in terms of restoration and work on those areas where we think that the thermal characteristics of the creek will support the native fish moving forward. So that's kind of the importance of temperature. And across all 4 tributaries, we monitored in-stream and sediment temperatures, as I said. So here the in-stream temperatures are blue and the sediment temperatures are orange. The sediment temperatures are taken at approximately 15 centimeters deep. That's to better represent the area where cutthroat trout might dig their redds and lay some eggs. And temperature is a big part of the egg development process too. So we figured this would be an important piece of data to collect. So in most tributaries, such as Burton and Plale Creeks, the sediment and stream temperatures only showed relatively small differences, typically less than 1 degree, suggesting that these creeks had limited thermal buffering capacities. But in contrast, Sharples Creek, particularly this station, it showed a very distinct pattern of the sediment temperature being typically on average about 3 or 4 degrees colder. So at SC 2, 3, and 4, we kind of cut off because of the screen sharing thing, but we noticed this pattern, so we ran a 2A ANOVA examining the effects of this on the stream, station, and logger type, so stream versus sediment, and their interaction was found to be highly significant in these reaches especially. Yeah, so just kind of playing around a little bit in terms of how significant the difference is in terms of stream and sediment. And that kind of points us towards the high buffering capacity of this area. Yeah, okay, so discharge. To measure discharge and stream flow in the 4 tributaries that I have laid out here, we installed level loggers paired with staff gauges and wildlife cameras. So this little bit of data you can ignore because the logger was kind of messed up and out of the water. But this allows us to calculate an estimated stream stage height at any given point in the year during the time that we're monitoring. So this baseline flow data has been also paired with site visits to get the actual cross section and flow of the stream on a certain day. So we're still building this data set so that we can get those rating curves just a little bit more robust and accurate. But this is going to help us understand how the hydrology and the flow of these creeks might change as we alter them with rehabilitation efforts. So here's a nice photo provided by Ben and taken by Christy, who works down at the Waldron Ranch. So this is the first, one of the first drone flights of Sharples Creek. So they would be the experts to ask about this stuff because I'm not a very in-depth drone person. But this work's being done by a master's student being supervised by Ben over here. So she's using LiDAR and ortho imagery, as well as I believe thermal imagery to assess the changes in the area that we're doing our rehabilitation work. So that'll be really cool to quantify the landscape scale change of the beaver dam analogs that I'm going to talk a little bit more about. So here's the first image that was provided to us, again by Ben. So yeah, this work is going to continue to expand as our rehabilitation within this reach of the creek also expands. I think currently we only have like 25 beaver dams there or something built, but we have plans to build over 100 in this small valley. So I think they're going to be returning in the spring to see how the first few beaver dam analogs have changed the landscape. But again, like I said, I'm not the expert on that part. So the fish monitoring stuff. So starting in spring of last year, we kind of, as a group, came up with a plan to monitor fish movement within and throughout the Callum Creek watershed. So like I said, we have 4 of these pit tag arrays throughout the watershed. We've got 2 on Sharples Creek upper and 1 on the lower portion of Sharples Creek. And then we've got a 4th one on Plale Creek. Okay, I'm not going to get through my whole presentation, but that's fine. So yeah, I'll just skip ahead a little bit here. Basically, this thing can detect fish as they swim through it because they have a pit tag in them. Here's another one. Yeah, here's the electrofishing reaches that we did. We electrofished around 12 kilometers, but a bunch of it we sampled twice, and it was more like 20 kilometers, and we did that over the course of 5 days, which was one of the more intense sampling efforts that we've ever done, but it was super, super fun. Caught a lot of cutthroat. Here's where we sampled. Sorry, I'm like speeding up now because I don't have enough time to get through all my content. So we sampled in this yellow region just once because we didn't really catch any of our target species, which is cutthroat trout. These green regions you can see on the upper sharples and the middle sharples. We had to sample those twice for the market capture study that was designed going into this to get that population estimate like I mentioned in the project's goals. And so in Prong Coulee, we sampled these two small reaches because typically this is where the creek is dry and anecdotally we've never seen fish here. So we sampled it just to test it and we did not catch anything. So we began with our restoration and lo and behold, a fish was there. So we're planning to sample it again coming up to add to the population estimate, although we don't anticipate catching very many cutthroat in that region. It was caught by hand, the one over there, by someone in this room. And same deal for Plale Creek. So we didn't really catch any cutthroat in this lower region. I don't think any, in fact, and they started showing up around somewhere between this yellow and green line. I wish I had a pin of the exact first cutthroat, which would have been great. But we sampled those green regions twice, again, to do the mark recapture study. The electrofishing was really tough and tight. You can see that it's like a crazy old Doug fir forest with tons of broken branches and people twisting and turning all over the place. So a lot of people in this room had a really fun 5 days of sampling in that. The capture efficiency was tough, so yeah. And then all the fish, like I said, over 80 mils got a pit tag inserted into them. So this is what allows us to detect their movements as they go through those arrays that we have set up in the field. So we had some professionals from the GOA as well as the U of C come out and help us and volunteer because they've tagged thousands of fish before and I've done like 5 in total now. So it was really, really big help to have, like I said, all of our project partners. The fish were let to recover for quite some time because they were sedated during this process just to make it a little bit easier on them. So there was no fish that ever got harmed during the tagging process, which was really, really great. So like I said, our target was to capture West Slope cutthroat trout in these creeks. And on the very first day of sampling at Plale Creek, we caught a bull trout here, which had never been documented in the Callum Creek watershed at all, ever. So this was like a really remarkable catch. So we gave him a little fin clip for the mark because we weren't allowed to pit tag this guy. It wasn't in our permit, even though that would have been cool. But then 2 days later, we actually recaptured him. So that was really sweet. We got like a perfect population estimate on bull trout. Yeah, and these guys, these cutthroat, they got pit tagged, like I said. This was one of the bigger ones we caught from one of the beaver ponds. So here is the preliminary results of the fish. This figure was also provided by Ben at FRI. And as you can tell, we caught significantly more fish in Sharples Creek than we did in Plale Creek. There we caught 1,565 cutthroat, and in Plale we only caught 68 cutthroat. And here is their length distribution, also provided by Dr. Ben. And a population estimate was provided by him as well. This is just preliminary stuff, and we think that there's around 2,200 cutthroat in Sharples Creek overall, but just 80 in Plale Creek. So you can see the stark difference between those two streams. No other population estimates were done. So here's some of our issues out in the field. A cow attacked one of our arrays. They broke the fence. completely got in there and ripped it apart. So we had to emergency shut that one off. Yeah, and basically the other issue was just like a lack of sun in these valleys. So late in October, the sun got too low in the sky and we lost power to it. So I'm gonna go really fast through this part. Okay, so this creek has a lot of good habitat, beaver dams, yada yada. We caught big fish in this area, so that's kind of what we're trying to mimic in our rehab. So these are some of the first few structures that we have. But like I said, we have plans for like over 100 of these structures in the upper Prong Coulee. And all those monitoring stations are going to help us detect the changes that are associated with these structures. Other things, culvert removal. There's 2 culverts up for the chopping block, and we're working with a ton of partners in the area for BMPs and other grazing practices to help the rehab area have even more rehab and healthy benefits. Okay, did I do it on time? Thank you.
Don't go anywhere.
Oh,
Yeah.
not done?
You're not done. Do we have any questions? You can.
Sorry, I can't. Oh, okay, Flora, go ahead. No, yeah, you're talking. And then you have back.
Cameras have any cell chips? Like how did
No,
you know?
yeah,
So
we
you
just,
had to go out there.
yeah, so the way that it worked was the camera is pointing at the staff gauge, and then there's also a level logger in stream. So they're basically like confirming one another. Yeah, so we had 2 years of that data, and they're never gonna like agree 100% because the level logger, it's detecting pressure, and then you convert the pressure into a height based on like how you install it. So they're kind of just to confirm one another. And then the flow, like, you know, a stage curve, like at this height, the creek probably discharges this much water. What we do to develop that is we visit the site on multiple different stages and collect flow discharge measurements with like a full transect, and then we can build the relationship like that. Yeah. At the back.
So you had, one of your charts showed that the difference between sediment and stream temperature.
Yeah.
Were you able to determine what was causing the significant difference in those specific?
Like not determine it fully, but based on all the work that we've done out in that creek, so those 3 stations are right in like the healthiest part of the watershed where there's tons of relic and active beaver ponds, like real big ones that are as tall as me or bigger. And so like the theory is amongst me and people I've talked to, we think that it's causing a ton of groundwater recharge and keeping the sediment temperatures a lot cooler. Yeah. So. Is that it?
Any final questions? No? Amazing. Thank you, John.
Thanks.
All right, so next up, we have one more presenter before our break. We have Noreen Ambrose from Cows and Fish. I'm going to try to get my process a bit faster here. Yes. That was fast, quickest transition yet.
Thanks, Casper. Thank you, everybody, for being here tonight. Can you guys hear me okay? Great. So most of what you're going to hear today from the other presenters is obviously pretty fish focused. That's why you're here, is about native trout. But I'm going to talk a little bit more about the other aspects of things that we might need to pick to work on to improve things for native trout. And that's not necessarily the fish themselves. So for those of you that don't know what a riparian area is, I'm sure you all do, but it's really that transition zone. But I also like to think of it as the gateway or the opportunity to connect land and water together, to the decision making, to the thinking, to the land use, to what happens in the water. And we know obviously fish rely on what happens on the land, not just what's in the water, and how it got there is kind of important to the fish. So riparian areas are that place that's, you know, a little wetter and than the aquatic, or pardon me, a little wetter than the non-aquatic, and obviously drier than the aquatic, that transition zone. And that's that sort of last defense less than 5% of southern Alberta being riparian. It's pretty important, and we expect it to do a lot on the landscape when it's healthy and functioning. What does that look like? It generally is going to trap sediment. It's going to resist erosion. It's hopefully going to build new banks as the stream naturally meanders, for instance. And it's going to support biological diversity because it's doing all those things, including supporting native trout, hopefully. But water has a lot of power, and that includes when it's in the stream, but also before it gets to the stream. And a lot of what we do on the land influences runoff, it influences soil movement. And really that's a lot of what we're talking about to benefit fish, aside from obviously angling pressure and hybridization and invasive species that are directly a problem for the fish. A lot of what we're talking about is really land use, not fish related activities, it's land use activities. So when we want to have a healthy riparian area, amazing, beautiful, complex, messy streams like this that are really hard to electrofish in, which is a good thing, how do we get them? A lot of what we might think about is thinking, how do we provide these things for these amazing fish and all of the organisms that are in the water, and ourselves downstream for that water quality we want. And a lot of that is, we all recognize that when streams are unraveling, when there's not much for deep binding roots, there's a lot of erosion, there's not overhanging cover, they don't have the habitat things that they need, and we're potentially actively contributing to reducing the quality of their habitat when the land use and streamside areas are not in good shape. And that, of course, comes from all kinds of land uses, both present and past. So it's not just what's happening today, it's obviously an accumulation of things from days gone by, as well as what's going on right at the moment. And a lot of these things, like these pictures, cows and campers are in the same place, quads and culverts, all of these things are adding on top of one another. It's this cumulative effects. And so, we, Cows and Fish, as well as many organizations, are doing restoration, where people are doing reintroductions, trying to obviously reduce hybridization. But how do we address these things? The cause or the root of the problem in these cases is not the fish, it's not the stream, it's the people that manage and use the land. So that's kind of what I want to focus on, is more as the human aspect than the fish aspect in the rest of my talk, and or with a lens to think about, this is about people, people who want to do good stuff, but also people that have influenced the landscape inadvertently or not, and therefore affected native trout. So a number of years ago, I realized, looking at this, it was almost 20 years ago that we did this. Some of you guys participated and helped in this. It's called the Mystery and Magic of Fish Survey. So we surveyed Albertans and basically said, what do you know about fish? It wasn't specific to native trout, but what do you know about fish? A whole bunch of sort of biological questions and a bunch of sort of attitude and knowledge and perception. And there's a section in the survey that was true and false, and we basically wanted to know if people could get the right answers. And what this question was, true or false? Native fish have been successful at adapting quickly to habitat changes we made. The correct answer is false. They don't adapt quickly. That takes evolution. So, but only 58% of people got that right. That's this number here. And interestingly, only urban and rural people, urban were slightly more accurate than rural. Recreational people were slightly better than that. Those in the government were slightly better than that, but only 74% of them participated, and 82% of nonprofits and volunteers got it right. And rural and all the three categories on the right, recreation, government, volunteers, and non-government, are in the urban and rural groups as well. So so there's a lot of people that don't know that the correct biology of fish, including people who are managing, and almost everybody that was participating on the volunteer and non-government side and recreation side and government side were all people who are interested in fish, whether they're anglers or they're managing fish or whatever. So there's a lot of misconceptions about what actually fish need, where they think they can get used to it. No, they can't. That's not how it works. At least not in the next few millennia. That's not how quick it'll happen. What they get used to is being gone, right? That's what we're seeing on our maps. So, but we also asked recently in our Eastern Slopes work about the importance and values that people had. And we said, like, what do you care about in Eastern Slopes? And one of the questions was this one, which was the importance of protection to you in different kinds of things. And one of the really important ones, which was the third one down from the top there, is, you know, you value protecting or riparian habitat for native fish species. And all of those, the top choices that people could choose, almost all of those high ones were ranked at 90% of people, whether they lived in the eastern slopes or were in the buffer, which includes Calgary, thought those things are all important or very important to them personally. So we know that these things are important to people. So how do we use that value that people place on things to actually affect change? We also asked in that survey a lot about what are you doing and what is your connection to the eastern slopes. For those people that were motorized recreationists, which I picked out here because that is one of these sort of extensive land uses that's not under, say, disposition in the eastern slopes. We asked them a bunch of questions about how do they use their off-highway vehicles. And on the far left it says, crossing rivers and streams on my off-highway vehicle is a normal part of my travel routes. And 43% of the people that live in the buffer zone, which includes Calgarians, said, yes, that's a normal part of my travels. 24% of people that live in eastern slopes normally go through the creek. But interestingly, at the other side of the graph, you see it's people that said, I only cross rivers and streams where there's a government-approved crossing, such as a bridge or a hardened stream bed. How many hardened stream bed crossings do we have that are on approved trails? Pretty few, which just means people think they're on a hidden approved crossing. And it's still showing that even though people value these places, they don't necessarily know what's actually happening. And we know from our other work that people don't understand fish biology. So how do we affect change? We know that healthier riparian areas, cleaner water is good for fish. This was some work that we commissioned a number of years ago in the kind of southern part of the foothills, sort of Todd Creek and some tributaries in that area. So sport fish, there's more of them when you have a healthy stream. And that's as measured through a riparian health assessment. All other fish are a little more tolerant, so there's not a significant difference in those kinds of places necessarily. So we know that the land is important to the fish. And it's also really important, one of the things we like to help people understand is that, you know, all green is not good, or in this case, all white daisies are not good. This is Lost Creek area, Carbondale, kind of south of the Crow's Nest Pass after the Lost Creek fire. It's not like weeds are only after fires, they're everywhere, but they affect the habitat because they affect the deep binding roots, they affect the filtering, the overhead cover. Daisies, by the way, there's no native daisies in Alberta that are white. They overhang, if they overhang into your creek, are they providing any shade or shelter? Are they providing deep binding roots and nice hidey holes? No. They're not providing structural material that falls in like a log. No. They're exactly doing all the opposite, replacing the opportunity for those plants. So understanding our land uses and our management choices in the case of getting weeds, rid of weeds, for instance, is understanding what a riparian area should look like, could look like, and obviously how to achieve it. So this is an example of planning a restoration project to say, if we understand the plant communities better, if we understand the complex land uses and the people that are using these places, we can hopefully build better projects. And this is an example of rough and loose on the far right, a red osier dogwood seed planting that's growing in some of that, and then just basically mapping, saying do this here, there, you know, a sketch map of what the color coding should be in different places. And but when we do these kinds of projects, they take a lot of money and they take a lot of effort, and they're only a small stamp of space. This is a picture from a project where a lot of these places that all the fine soils and the nutrient-rich soils are gone. They went into the creek and down the creek. So planting things is tough when there's only rocky soils, and that's what a lot of our foothills are made up of, lots of rock and gravels. So, you know, this is an example of 50% survival rate, and there's lots of water. It's not necessarily overly dry in this site, which some of them are too dry. It's just really hard to affect big change when it costs a lot of money and we can only do small spaces. So how do we actually affect bigger change? It's not just restoration, right? We have to actually focus on changing management, changing land uses, and knowledge and attitudes are what get us there. So how do you know what you need? Obviously you have to understand the site well, you know, whether that, in our case, we're doing a lot of work on the riparian area, understanding what the plant communities are, understanding the management that's happening at these sites. And I mean, it sounds really simple probably to a lot of you, but how do you promote and support deep binding roots? That's what we need on the banks, on the floodplain. So how do you? And you don't get to plant them. How do you do that? How do you prevent the damage?
I wouldn't say restrict use, but manage use.
Potentially restrict use, manage use. How do you manage use? Educate. People care. People value.
Providing good alternatives to recreation.
Provide good alternatives. Yeah, like I think, you know, I want to think, I value the ecosystem and the fish, but I know it's us as people that make the choices. And, you know, I'm one of those people too. So how do we affect change? We know people value these places, but we also know their knowledge isn't as good as it could be in all walks of life. And we also know that the uses are still happening that cause that cumulative effects to continue. So helping people understand that cold groundwater comes from somewhere. It had to get stored first. A healthy site slows it down, lets it seep back in. Maybe it's in a beaver dam. Maybe it's something else. It's finding these opportunities to support, you know, livestock producers use off-site watering systems and electric fencing to redirect use, limit use, building bridges so people aren't crossing through the creek, whether that's the right kind of bridge, the right place. Obviously, one of the ongoing challenges is needs keep changing. Recreational vehicles keep changing. They're wider. This bridge hardly fits anything now, except a basic old quad maybe, and a bicycle, and a foot traffic. Compared to a lot of the side-by-sides, right? They're getting bigger and wider. And restoration can have a real positive impact, so I don't discount it. And I think one of the important things is getting people to be part of that process. So they take ownership of, and a blood, sweat, and tears putting it in also helps them take ownership and tell their friends, hey, I did that. Let's keep doing more of that, and I don't want to see it wrecked. And I'm going to tell my friends, let's go keep supporting that kind of work. We know that changes can happen when we make, you know, grazing changes with livestock producers. This is a place that has bull trout. It doesn't have to be exclusion either. This is an example where grazing is still happening in this decadal period. It's just how it's grazed, how it's managed. So alternatives are really important. But I would say one of the things we often get asked is like, what are they? Was solution is to put up a fence, or the solution is put up a bridge? No, the solution is how do we achieve a healthy ecosystem? We want to focus on the outcome. Deep binding roots. We want to focus on things. What are the 4 principles of grazing management? I work with lots of people with grazing. Balance, distribution, timing, and rest. There's no one right way to do that. There's 1,000 right ways. Same with how do we provide what fish needs, reducing sediment, providing cover, shade, and structure. And obviously water, because they need water. Weaving those together between land uses is critical if we want them to succeed, and especially in the busy eastern slopes where we have them all on top of one another, these different kinds of land uses. So finding the principles of different land uses and finding ways they can complement them through the people that are involved. Many of you have been or heard of the Buck for Wildlife projects in the Rocky Caroline area, and this is pictures from some of that work long before my time in terms of working on these issues, but they also show it takes a lot of time for change to happen, and we don't have time on some of these places. They're blinking out. So we need to affect big change in big places, in big areas, and tackle it lots of different ways, which is about working collaboratively with different organizations and entities and folks like yourselves, as well as the public. So I would encourage you to do that, and I would really encourage you to think about sharing your practical knowledge as well as your passion. One of the last surveys we did is, again, understanding people. People are more likely to listen to you if you show your passion. Share your, and I don't mean your romantic passion, show your passion for the environment. Show your passion for these amazing spaces, places, and wildlife, including native trout. It does affect their learning from you, and it does affect their willingness to make a change if you are committed and enthusiastic and share too. If you just tell them things, that's monotic and not that interesting, it's not that interesting. Why should they care? Your passion matters, so share it. Thank you.
Excellent. Thank you so much. Do we have any questions in the audience?
Yeah, how many do we, I know, I was going to look that up because it's not on the slide, but I think it was over 200. So it was quite a lot. It was statistically significant. It was considered an adequate representation for the population of Alberta that lived in that zone. So it was, and it was conducted not by us, by third-party social scientists that we hired. So.
Yes, question over here.
No, not a question, but I want to thank you for your passion. Your initiative in the recovery and conservation of native trout. Predates this collaborative and many other forms over 10 years ago. So thank you very much for it.
Thanks.
No more questions? Amazing. Thank you so much again. All right. So that concludes our first 3 presentations. We are going to take a 10-minute break. We're going to take a 10-minute break. So I'm going to mute our folks online, and then everyone else, feel free to grab some snacks, go to the washroom, and we'll be back here at 3:30. Yeah.
Be careful this time.
And the folks online hear me again? Just give me a thumbs up or a message in the chat.
Sorry, I don't want to get blinded.
No worries. That's good. Excellent.
All right, let's sit down.
Mouse. There it is. Okay. Excellent. We are going to get started right away here. So next up, we have Ben Kissinger from FRI Research.
Super. All right, everyone's in. We'll get rolling. So I was asked to talk about things that would have application or could be used as tools. And so I'm not going to talk about a ton of the science I'm doing right now, and it's more focused on some products that people might be interested in utilizing in day-to-day operations. And so a question that we get often is, who is FRI? Our headquarters are not based down here. We're actually out of Hinton. And FRI used to stand for Foothills Research Institute. We are now an acronym so that we can work outside of the foothills, although we still do a lot of our work in the foothills. And it used to also be called the model forest, so a lot of the work was forestry focused. I have a little sub-hub down here in Calgary where I'm an adjunct professor at the University of Calgary, but this is my team. And so you might look at it and be like, there's not a whole lot of people there. What we rely on heavily is collaborations. You've probably heard about some of them already today, but we can't get what we do done without collaborations, without undergrads. And so, yeah, our collaborators are the seventh person on the ice. Our study area spans predominantly the eastern slopes, which is Treaty 6, 7, and 8, as well as home to the Métis Nation of Alberta. And we try our best to engage and oftentimes collaborate with communities along the eastern slopes in hopes of reconciliation. We talked about a lot of this earlier. This was a diagram that I had in my undergrad in the US describing what influences fish abundance, and it's quite simplistic, and many of you would say you're missing stuff, which is true. And so how fish abundance is influenced is through cumulative effects. And so to try and understand what is the most important one or suite of important ones, you need to have good data for it. And I think our work in the Kakwa watershed is a great example of why cumulative effects are important to look at. So if we look at the top panel, that's linear features in that given watershed. In the middle, it's forest cover. And so the areas of lighter colors would be timber harvest and then regeneration back to a mature forest. We have spills down in the bottom or middle panel, which is pretty intense in certain areas, as well as temperature. And from like a modeling standpoint, you'd like those to happen evenly across the watershed, but they don't. And so you have to account for not only the natural ecology of the species, but also where these impacts are happening to be able to try and understand what is causing declines in particular species. So I'll jump through this real quick. We have native trout, they're not doing well. For this particular one, I'm going to focus on bull trout just to have a consistent species as I talk about water temperature. So water temperature is super important for all of our native trout species because they're exotherms, and the water temperature surrounding them influences all of their bodily processes and ultimately how much they grow in somatic growth, as well as their abilities to reproduce. And because of this, they're highly associated with certain temperatures, especially our cold water fish species. And because temperature is influenced by a range of factors, including precipitation, solar radiation, vegetation, groundwater inputs, and in Alberta, a lot of snowmelt and glaciers, it's really important to go to many places on the landscape and measure temperature. And so we do this using small hobo-tibbet loggers, which are in the upper right-hand corner, to be able to understand what temperature is at a given location. We then get these outputs here. We have two different streams that were monitored, one in blue and one in red. Jonathan was talking about these earlier. And basically, we have an hourly record of what temperature is there. And when we understand how a particular species grows,
hopefully I gotta stay over here because you need to hear me.
You can use the little.
Does it work?
Yep.
We see that for bull trout, as we mentioned earlier for West Slope cuts, there's an optimal range of temperature at which they grow at. So this is their growth rate peaking somewhere between 10 and 15 degrees Celsius. So if we overlay that on these temperature profiles, we see that this blue stream is cool enough for them to exist in throughout the entire year, whereas the red stream exceeds it. And because of this, if I had to put my money down, I would bet that bull trout are more likely to be in the blue stream than the red. It also could mean that bull trout need to move in and out of this stream. So maybe this stream is only functional for them in the winter months. So to understand that, not only at given locations, but for the entire eastern slopes, we created this collaborative approach of putting out temperature loggers on the landscape to understand what water temperature is. And we went from approximately 700 unique locations in 2023 to over 3,000 now. We had contracts, including with FCC, Sinowitchi-Winiyak Nation, Swan River First Nation, and others to deploy temperature loggers. But our biggest bang for our buck was just talking to people. Many organizations have been collecting this type of data already, and were willing to share it with us to bring it into this larger model. And so, yeah, this is just kind of a timeline showing how we got to where we are today. So yeah, 3,000, over 3,000 unique loggers, some locations spanning 30 years of data, and over 110 million reads. And where we want to get to, and we are so close, and I wish I could have shown you a figure of a model like this, but is taking small scale Huck 8 watersheds like this, the Red Willow Beaver Lodge watershed, and having it for the entire eastern slopes. When we have this type of data, we can then talk about where thermal habitat is available for our cold water species. So here we look at the month of August because it's typically the warmest month with the lowest amount of flows. On this side, we have all water temperatures ranging from 0 degrees Celsius to 22. The loggers are in blue, and so the warmer colors here along the main stem are warmer water, whereas the cooler colors are cooler water. So if we restrict it to just the temperature range that bull trout likes to exist in, 15 degrees or less, we see we lose many of those rivers within the given watershed. So this kind of tool over here can be really useful for managers to say, where should I prioritize my efforts? Should I be able to see species X, Y, or Z in here? And if not, why maybe we put our effort in this place or the other because resources are very, very limited. So that's one tool that we're working on. Another one that's really important is stream networks. So in that previous figure that I showed, if there's any hydrological modelers in here, having a continuous network of rivers is really important for doing any models because streams have directional flow. And so for temperature, they typically get warmer as you go downstream. So having a continuous layer is essential for doing those models. When we look at the FWMS hydro arcs, they're actually in these grids, and they're pretty accurate because they work off of satellite imagery. But because they're grids, the second you hit the edge of one of them, you can't model anymore. So the other option is to use create synthetic networks using digital elevation models. But sometimes in low resolution LiDAR, you get pretty variable data. And I put this up here. Sorry, I didn't have another example, but where the stream wouldn't land where the stream actually is. And this is problematic, especially for people doing management, like hypothetically forestry that would have a certain amount of buffering surrounding a river. If it says the river's up here and that's critical habitat, it might be very far away from where the river actually is. So having an accurate stream layer is very, very important. So we've partnered with TerrainWorks to build these higher resolution networks, leveraging data that exists within a bunch of the forestry companies' FMAs. So forestry companies are very interested in trying to understand the height of trees that exist, as well as the species composition. So they have higher resolution LiDAR that can then be incorporated into developing much more accurate stream networks. Another cool thing about these stream networks is that we can get other important covariates. So we get estimates of wetted width, which can be used for management purposes. From a forestry standpoint, we get increased accuracy. And then from a fish standpoint, we get contributing area, gradient, width again, the floodplain. So a lot of other neat covariates that can ultimately be put into models that help predict where you might see certain species on the landscape or stream temperatures. So this is another tool. We are almost done. Unfortunately, we just added sundry, so it's going to take a little bit longer to fill in our map, but we've completed first versions of this network for the rest of the FMAs that you see in watersheds here. So this will be a helpful tool, hopefully, for people to use to get more accurate data or use in modeling purposes. So the last thing that I'm going to touch on today, super, I got 5 minutes, is a project that is led by Dr. Angus Lothian, a postdoc in my lab. So I guess I will talk a little bit about science. But this kind of brings together considerations of those various land uses, cumulative effects, and how it might be shaping what we see on the landscape. And so this is a recent publication. It also links in a little bit to what Mike was talking about earlier. Here we're using occupancy models versus abundance models. And the reason we're doing this is we actually have quite a bit of data in Alberta that has been collected over time. So this is the spatial distribution of river and lake data. And so we have pretty good coverage. The tricky thing is multi-year data. We don't have a lot of multi-year data. And so Angus went through an exercise and was able to identify 74 Huck 10 watersheds along the eastern slopes where we had at least one measure per year within a 5-year bin period between the years of 2001 to 2020. And by doing this, we could look at how species presence absence changed. Another neat thing that we did was we accounted for regeneration of forest as well as restoration of linear features within our watersheds, because a clear cut doesn't function as a clear cut 20, 40, 50 years after the fact. So I think that's a little bit of a novel component with this approach. What we saw was in those 74 watersheds, and all of our observations are going to be at the Huck 10 scale, so don't be like, oh, this is site specific. This is at the Huck 10 scale. We saw that tree coverage decreased significantly. Linear features increased, but was not significant. And so the key findings are what species were changing in occupancy in those 74 Hucks. And this is 74 Hucks, not all watersheds on the eastern slopes. And what we saw was that bull trout had, so this is a negative change in occupancy, whereas this would be positive. So overall, our native species were on the negative side of the spectrum, whereas our non-native species were either straddling no change or a slight increase. And for this, because this question got asked earlier, how are we able to genetically tell fish apart? We weren't. So this is purely rainbow trout in the Athabasca drainage, and this is West Slope cutthroat trout in West Slope cutthroat trout's distribution. So while they're straddling 0, that doesn't account for all the populations that are hybridized or introduced. So these values are likely lower than that. This is purely working off of the FWMS data that existed. So keep that in mind for the rest of the figures here. So some general trends that we see is that our native species tend to exist in areas of higher elevation. So this is the probability of occurrence on the y-axis, and then whatever factor we're looking at. So this one's elevation. Athabasca rainbow are a little wonky, but we feel like that's because of stocking, maybe contributing to this little uptick there. Whereas all of our non-native species tend to be at lower elevations. When we look at linear features, we see bull trout at a pretty strong negative relationship. And the others, it was a little less clear, but typically negative. Oh, did I go in the right direction? I skipped one. Forest cover. Bull trout or our native species had a positive relationship with forest cover, whereas our non-native species were either stable or negative, stable. Sorry, I had that in there and I must have just deleted it. a big thing to know with these data here are, we're looking at this snapshot in time. We have 20 years, and that's a long amount of data, it seems like, for people who work on these types of projects, but we don't know what it was in the past. So the trends we're talking about here are in the past 20 years. So just keep that in mind when thinking about these data outputs. Another important thing to think about is the data that we're looking at is associated with occupancy relationships with some factor. It's not change. So bull trout and their positive relationship with forestry reflects their ecology as well. We typically find them at higher elevations, cooler waters in more forested areas. So if we want to look at change, we need to measure over multiple time periods at the same locations. And that's where the CACWA study comes in. I can't get into it now because we're not completely finished, but here we have two time points, as well as being able to account for how linear features, how forest cover changed over that time period. So I'm really looking forward to presenting that in the near future. So some general conclusions: cumulative effects are tricky. Interpretation is often influenced by available data. Hopefully some of these new tools we're developing can help managers and land users. It's really important that we account for natural ecology of species when trying to talk about what's causing change, and hopefully more results to come soon. Big thanks to our funders, our collaborators, the Temperature Project, and the Stream Network were funded by FRIA. And at this point in time, I'll open it up for questions.
We don't have any questions in the chat. So do we have any questions?
Yep.
You mentioned you only have 20 years of data. Have you ever considered using backdropping?
Yeah, that's a great question. So we actually have a project that started up this year in Swan River watershed with Swan River First Nation, and we're using that to kind of think about how the FSI has worked and see if we could incorporate traditional knowledge in Arctic grayling within that area to get a better understanding of where they might have been historically. So yes, we're trying a pilot study right now, but I think that is a great approach and probably something that would be beneficial within those FSI models. Jess?
In terms of the grayling range, do you think there's a possibility there's some undiscovered temperature data sets out there, or is that like a true gap?
Temperature data sets?
Data sets, yeah, because it's
Yeah.
very gappy.
Oh, yeah, no, there's not. Yeah, so I mean, if people want to pay me to go up and put temperature loggers up out north, well, that'd be great. We do have some surveys we'll be doing in the next kind of decade up there, and we'll be incorporating temperature monitoring into that approach. Yeah, the approach we're doing for temperature has been done for the entire western half of the lower 48 states. So this is a really powerful tool, an important variable for these fish that hopefully can get created for Alberta. Elliot?
Is there any like opportunities to like cross-pollinate like some of the like Dan Isaac, climate shield, and all that kind of stuff?
Yeah, so the model we're running is the same one Dan runs, and the hope is that it is then functional and comparable across the border. That's why we've put in as much effort we have. Jess?
I'm not sure if you can speak to this right now, but do you have plans for the next step with climate?
Yes, we do. And so we have the data sets in place. We're building the static model first, and then after that, we'll incorporate the climate variables and have projections into the future. So yes. And honestly, I could have an email in my inbox saying, here's the output for version 1 right now. We're like, we have the points on the map, which is the hardest part. Now we just need to get the model finished. And by we, Mac Hydro with Ryan McDonald is leading that effort, so I don't want to take all the credit there.
All right, thank you, Ben.
Yep.
So next up, our next presenter is Caitlin Gifford from Alberta Environment and Protected Areas.
even though I'm high maintenance already. Okay, hi everybody. My name is Caitlin Gifford. I am a fisheries biologist with the Government of Alberta. I work within the Fish and Wildlife Stewardship Branch in the Ministry of Environment and Protected Areas. And today I'll be talking about native trout recovery stocking, which is stocking within the conservation and recovery contexts. And before I begin, I just want to take a moment and acknowledge that fish stocking has a long and complicated history in Alberta. As native trout and fish enthusiasts in the room, if you hear fish stocking and some alarm bells go off, I'm glad. And let's talk about it. And there has been historical widespread stocking across the landscape, and it really has contributed to the significant declines of native trout. However, when used intentionally and in the right places with thoughtful planning, stocking can be an important conservation tool that's going to help us address recovery challenges that can't be solved through habitat work alone. And with that, let's begin by considering some of the problems or threats facing native trout in Alberta. So native trout are impacted by a combination of cumulative threats from what we call the 3 H's. So harvest or overfishing, habitat degradation like sedimentation and fragmentation, and hybridization or competition with non-native trout. And understanding what's driving declines behind native trout by identifying the specific threats to a population is the first step in any recovery effort. An effective native trout recovery involves reducing and in some cases even eliminating those threats. This might sound a little bit straightforward by how I've described it, but as Ben stated there, cumulative effects are tricky. This is tricky business. So once we've identified the threats affecting a native trout population, the next step is choosing what the right recovery tools to address those threats. And recovery actions are underway across the East Slopes utilizing these tools and more, and those are part of a coordinated effort by the Native Trout Collaborative and beyond to address cumulative threats to our native trout populations. And there is some encouraging news. Recovery actions have the potential to improve the risk ranking of roughly 65% of native trout populations in Alberta. These efforts can have a meaningful difference. Zooming in on recovery stocking as one of these tools that operates within the greater system of recovery actions, let's keep in mind that recovery stocking isn't a quick fix, and it isn't a standalone conservation tool. So in very specific situations, recovery stocking can complement other recovery actions like habitat restoration, connectivity improvements, or non-native species management. In other words, the declines of native trout aren't a problem we can stock our way out of. Oops, sorry. So what is recovery stocking? This slide shows a visual example of what recovery stocking can look like. So in this example, eggs and milt are collected from a wild native trout population. This might be a nearby or nearest neighbor population. Those fertilized gametes are transported to a fish hatchery where they're temporarily incubated. And then once they reach the appropriate stage, those developing eggs are transferred into a remote stream incubator where they finish incubating and hatch directly into the natural environment. But this is just one example. The recovery stocking process can look different and vary depending on the species that we're working with, whether it be any of the native trout species we've talked about today, the project, or the specific conservation objective. However, across all projects, the principle is the same. So it's using stocking as a conservation tool to reestablish, expand, or supplement native trout populations. And it's also important to note what recovery stocking is not. Recovery stocking is very different from the historical stocking practices of the past, and it's also not modern day recreational stocking, which is stocking for the primary objective of either creating or enhancing angling opportunities. So for recovery stocking, there is great attention spent to preserving the genetic diversity in our native trout populations, maintaining local traits, genetic traits, and minimizing artificial selection and hatchery pressures. And why do we even need to consider recovery stocking as a conservation tool? If we look specifically at West Slope cutthroat trout, the non-introgressed populations, which you may know them as pure populations, are the green stream sections in this map. And they're now fragmented to the extreme headwaters of the east slopes. And these populations occupy just a small fraction of their historical distribution. Looking at the yellow and orange stream sections, those are populations with increased levels of introgression or hybridization, and they are a bit more widespread across the native range. So these pure or non-introgressed populations are the last refuges of non-introgressed West Slope cutthroat trout, and they're incredibly important. So their genetics represent unique local adaptations, traits that were shaped over thousands of years to fit the conditions and the variability in these headwater environments. Noreen mentioned evolution. These fish have evolved to live here and thrive here. And these adaptations are critical for the long-term persistence and resilience under natural and changing environmental conditions. So the reality is that addressing habitat threats, which is essential and remains foundational to recovery work, it will not on its own restore resilient, genetically intact native trout populations across the landscape. And this is where recovery stocking becomes a necessary tool. Double tapping. Hope I didn't miss any slides there. Okay, so let's zoom into some details behind how recovery stocking is actually applied. So recovery stocking requires intentional application. This means it has to be purposeful, thoughtful, and grounded in evidence-based planning. And that planning starts with asking ourselves, what problem are we trying to solve? The answer is firstly going to tell us, is recovery stocking an appropriate tool? And if it is, what approach or category of stocking might we consider? And there are 4 categories. Each serve a different recovery purpose, comes with its own benefits, risks, and considerations. So a new introduction is introducing fish into habitats within their historical range, but where they currently don't live. So these habitats could be historically fishless waters, say above a waterfall, or they could just be habitats that don't contain hybridizing fish species. So we wouldn't want to stock rainbows, Athabasca rainbows, or West Slopes into waters that already have rainbow trout, as an example. So an example of this would be replicating a native trout population on the landscape above a barrier to build resiliency. Next, we'll look at genetic swamping. And so this can be used where historical stocking has introduced a very small amount of introgression or hybridization into a population. And on this slide, that's represented by the purple nose on the first rainbow trout there, or on the first West Slope there. Adding a large amount of fish that are not introgressed, they don't have a purple nose, via swamping, can shift the genetic threshold to reduce overall introgression, or the amount of overall purple nose in this example. Next, we've got genetic rescue. So some native trout populations now have to be managed for isolation. And isolation can lead to very low genetic diversity or even inbreeding depression. And recovery stocking here would involve what we call assisted gene flow. So moving fish or gametes into these isolated populations to try to increase genetic diversity. And lastly, we'll talk about supplementation, which may be considered when a very small population is at risk of collapse. And critical intervention is considered to give reproduction a short-term boost. Supplementation must be used with extreme caution. There are serious risks to genetic homogenization and population level effects. Supplementation at its best is like a Band-Aid, unless we're actually addressing the underlying threats behind declines. And so it must be paired with other recovery actions to support long-term population viability. If this sounded like a lot, I'm not a conservation geneticist, so don't worry, there's no test at the end, but just to try and give you an overview of some of the complexity. So this intentional application of recovery stocking is just one part of the detailed multidisciplinary planning process. And that process draws on a wide range of expertise. So that's conservation genetics, fish pathology, biology and ecology, and of course, fish husbandry. So the source population and the recipient population or habitats are thoroughly reviewed for genetic compatibility, pathogen and parasite risk, habitat suitability, and more. And careful consideration of each metric is critical and starts to shape what a recovery stocking project could look like logistically and operationally. And let's just talk a little bit more about genetics, which provides a roadmap for recovery stocking. NTC partners and collaborators have collected thousands of fin clips from native trout populations across the East Slopes. And this genetic data set is helping us answer questions like which populations are not introgressed, which are most unique, which are most at risk, and where our recovery actions can have the greatest conservation impact. So genetics is helping us avoid harmful decisions during recovery stocking and supports actions that actually maintain or even improve adaptability and long-term persistence in native trout populations. And the reason planning is multidisciplinary is because this tool comes with risks that must be carefully managed. Poorly planned or poorly executed projects can cause a number of unintended consequences. We can genetically homogenize populations. We can lose those unique local adaptations that are so important for long-term persistence. We can transfer disease, we can disrupt ecosystems, and we can waste the efforts of the Native Trout Collaborative and partners. If we aren't careful, we don't only risk project failure, but we risk harming already imperiled populations. And so that's why it's so important that these projects are thoroughly and rigorously planned, and that we work to minimize threats before recovery stocking occurs. And these projects aren't theoretical or experimental, they're already happening across the landscape, across North America and right here in Alberta. So many partners and organizations are all contributing to these hands-on recovery stocking projects. And these real-world projects give us valuable insights. Collectively, we are learning what works, when to adapt or refine approaches, and where we can apply similar projects in Alberta. And we'll just wrap up by summarizing a key message from today, that recovery stocking is a critical conservation tool, but it's only one piece of the recovery puzzle. Stocking alone won't achieve resilient, self-sustaining native trout populations, but as part of a coordinated recovery system, it can play an important role in moving us towards this ultimate goal. So that brings us to the end. Thank you for listening, and thanks for everything that you all do with native trout recovery.
Awesome, thank you so much. We don't have any questions in the chat yet. Do we have any questions in the room?
Yes. What's that average time frame for a recovery stocking project?
Yeah, it can vary. Excuse me. I would say it's reasonable that projects take multiple years to plan, to collect the data we need to understand the risks and the benefits of projects. And then once planning is completed, it's likely multiple years of recovery stocking. So these are kind of projects that reasonably take 5 or more years, 5 to 10.
Do you always grab the eggs and the milk from wild species or wild capture fish, or do you ever take a brooding stock and keep them in captivity for reproduction?
Yeah, both can be done. So they're looking back 5 years or so. There was an intensive amount of work done to create a old man watershed composite broodstock. And so there is a broodstock available at the Allison Creek Hatchery that was sourced from wild populations of West Slope cutthroat trout in the Old Man Basin. And so that's used for recovery stocking purposes as well as recreational stocking purposes to kind of reduce the risk of stocking in some recreational areas that anglers like to fish. And we can use this broodstock to instead of rainbow trout or other West Slope cutthroat trout populations. We try and pair the source with what's most appropriate. So sometimes the Old Man West composite broodstock might be an appropriate source, but if we're working in the bow, maybe it isn't the best source. So that's where a wild or a nearby population might be more appropriate.
Is there any opportunity for stocking hybrid non-native species that helps bring back the native species? You know, like mix, I guess, in a way, so that you're decreasing the reproductive capabilities of the non-native population.
So I think you said hybrid non-native populations. Is that right?
Well, yeah, I guess I mean stocking non-native species in a way that helps reduce those populations.
Yeah. So an example that I can think of is what's called Trojan male brook trout. So these are sometimes called super males. They're brook trout that have 2 Y chromosomes. And so if you stock these Trojan male brook trout, the offspring are all one sex because they're double Y chromosomes. So that's an option. It hasn't been done in Alberta. It is used in the States in some places. It's never been tested in areas that overlap with bull trout. So there's lots to be learned still where bull trout habitat and using these super male brook trout. But where brook trout don't overlap, it is an interesting kind of innovative tool that I think there's a lot of great science to demonstrate how it works and that it actually does work in reducing brook trout populations.
One more quick question, and
Sure.
then we'll move on.
Thanks for your presentation. One of the slides shows Parks Canada's projects. To what extent does the government of Alberta collaborate with Parks Canada, either methods or source populations or methodology?
Yeah, I would say we work quite closely with Parks Canada. They have wonderful programs for recovery stocking and they're making great gains. I talk to Parks Canada colleagues many times per year to hear what they're up to and how they are learning and advancing their programs. So in terms of source populations, there's certainly the potential that they would offer up some of their, once they've restored some populations and they're available, we could consider actually using them outside of national parks. An example also is they have explored using a different egg incubation technique. I described remote stream incubators in my presentation. They use a different technique called Jordan Scotty egg boxes that seem very functional and maybe a little bit easier to operate. So we're excited to use those in Alberta, and I think we'll be using them this year actually.
Thank you so much, Caitlin.
Thank you.
Awesome. All right, so now it's time for our final presentation. So Angela Ten from Freshwater Conservation Canada will be presenting. I will get it up.
Okay, hello everyone, my name is Angela Ten. I am a biologist with Freshwater Conservation Canada, And today I want to talk a bit about our restoration project at Radiant Creek. It's been ongoing since about 2020 now, so it's pretty cool. This is like quite a long running project, and for me, quite a personal one because I was there when it started, and I'm still here now that it's starting to like wrap up and evolve. So really cool to get to see a project go from start to finish. Radiant Creek is a tributary of the Clearwater River, so that's in the North Sask system. In terms of native trout, the only native trout that are there are bull trout, but there are also non-native brown and brook trout, and as well as a host of other native species like mountain whitefish, dace, all that sort of fun stuff. The big problem with Radiant Creek is that it has been channelized in size. So you can see here that this blue line is still, for some reason, what FWMS thinks Radiant Creek looks like. This is the old hydrograph, but it seems like sometime over like, maybe there was a huge flood event, and because this is the trunk road here, it's like the creek is being constricted by a bridge, and I think there was probably a big flood, and all the water got constricted here. It had a lot of power, and it just kind of like shot out and turned the creek super straight, disconnected it from all these old meanders, and it lost a lot of length. It is now like very disconnected from the floodplain, so it had a lot of problems with its like riparian and aquatic health. Here's what it looked like on the ground before we did anything. You can see that the creek is like relatively featureless. It is just kind of like a straight thing. It's pretty shallow throughout. It was really over widened, and it was really overgrazed at the time as well. You can kind of see here the grass looks bad. And there's like all sorts of cliff shear and all sorts of stuff on the creek, depositing a bunch of sediment. There's like so much erosion going on. There was lots of poop in the creek. So it was just altogether like not a great situation going on at Radiant Creek. So the big problems at Radiant Creek was just the lack of structure. It was kind of stuck in this degradation loop because the creek is so featureless and like there's nothing to provide it any structure. It couldn't really kick itself out of the degradation that it was experiencing. So in order to address that, we came in 2021 and added a bunch of PALs. So these are post-assisted log structures, is what the acronym stands for. They basically mimic accumulations of woody debris in a stream, which are very common in like this type of like mountain foothill area. And they serve to add structure to a stream to force it to do things that are not just move water. Here is a cool picture of some of my colleagues back in 2021 building one at Radiant Creek. And this is kind of a diagram showing how PALs work. So if you have the flow of water flowing laminar in one direction with no interruptions, when you add a PAL, and this is also the type of like mechanism that happens when you just have like a down tree falling into the creek, it breaks up the flow of water so it's not all flowing in one direction. It creates zones where the flow is constricted and flowing faster, zones where the flow is hitting something and slowing down, zones where the flow is dispersing and slowing down, and all that creates a bunch of heterogeneity in the stream. It creates scour pools like this because of the constriction jet like digging out the substrate and causing a bit of a pool, a bit of a fast moving area. It creates some erosion. The erosion of the banks, all the sediment is then deposited in the slowed down area to create new gravel bars and create kind of like a diversity in the stream bed profile. So it's not just all one depth, it's got like deep parts and shallow parts and all that. And just altogether forms a bunch of structure that creates good habitat, because these kind of like straight flowing streams like this are very efficient conveyors of water. But that's not what fish need. Fish need like diverse habitat, and that only happens when the water is not moving efficiently anymore. So we built 12 structures downstream of Highway 40 on Radiant Creek throughout about 2-3 years of time. We built a whole bunch at first and then kind of came back over the next 2-3 years to add on top of them, move them a bit, edit them. And these are a few photos of some of the PALs that we built. We were super fortunate as well that, let's see if this will play. We built them in 2021, and in 2022 we had like a crazy flood. Like it washed out sections of like Highway 752. The flood was so insane here. And PALs really do their best work at high water. The other crazy thing about this flood is, I don't know if you see the time lapse, this is over like 48 hours. Like the water came up like crazy. And that's really great because PALs do their work at high water. That's when like most of the water and sediment is being carried, being influenced by all the woody structure, and then forming new habitats. So here is another cool example of how, just kind of showcasing what I mean when I say PALs do their work at high water. So pay attention to the stream center line. This is where it's flowing at first, down here. That's the Thawag. This is like before any flooding happens, and this is all with one structure. So the water really comes up, the floods happen, and like where the water flowing changes. As the flow of water drops down a bit, you can see that the center line has changed. It used to do this, but now it's doing this. And it's all because of the influence of this one post-assisted log structure. So this is kind of the potential that we saw in the project. This is what we were hoping it would do when we started doing this stuff, and as you can see, that is what it's doing. So here's some pictures of a structure on the ground. This is what we call a mid-channel PAL, so that's one that's kind of like not associated with any bank. It's just placed in the middle of the channel. It's meant to mimic like a fallen tree that is just kind of stuck on a gravel bar. That's the type of thing it's meant to mimic. So here's what it looked like when we installed it on the left in 2021. Here's what it looked like during high flow in 2022. Not at super high flows because that was like legitimately dangerous to be around. This was a little lower, but it's like around the same time. They were both around June or around July. And then here's what that same structure looked like in 2023. So a lot of change has happened. I would say this is about a similar flow state as what it was like in 2021 when we built them, but you can see it looks super, super different. This structure is really cool too, because when we built it, we kind of were like, maybe it'll do something. We were like, let's just do it. It doesn't really hurt, but it has probably experienced the most change out of any of the structures that we built. So it's a super cool one that I like to bring up a lot. Here is kind of a comparison side by side of what it looked like in 2021 and then what it looked like in 2025. So super, super different. There's so much stuff racked up on it now. And 2025 is like a little more high water, but you can still see the change that has happened. Zooming in just on what has changed in 2025, first of all, you can see there's like a whole new bank that's formed on the one side of the PAL. It's no longer a mid-channel PAL, it's pretty much a bank attached PAL because the work of that single PAL has caused over a foot of aggradation. Like all that gravel has kind of deposited out and formed a brand new gravel bar, a brand new bank. And all that bank was created from the sediment shadow of a single PAL, just from the influence of this single structure slowing down and disrupting the laminar flow of water. There's a new bend in the channel. This section is also now a knee-deep pool. It was kind of freaky when I went there the first time because I didn't think it was that deep, and it was. There's fresh new gravels in the channel. These gravels are like perfect. They're clean. They're great for building reds, not at this specific spot, but that is like throughout the whole treatment reach where we worked on. There's a bunch of woody debris. There's a new undercut bank on this side of the creek. It's great fish habitat. Every year that we fished it since 2021, there have been fish there. Just last year when they fished it, there was fish associated with the wood and the bank. So it's clearly great fish habitat. And there's like brand new willow growth. The riparian health is phenomenal. There's like, this willow was not there when we built it in 2021. This thing's like 4 foot tall, probably more at this point. And that's all just from a few years of rehabilitation. And also we fenced the whole project area out to cows. So lack of grazing impacts as well. Here is kind of the same, this is a different part of Radiant Creek, but I wanted to show similar things just from a different point of view. This is aerial imagery. We've been doing aerial imagery here for 5 years. So here's what it looked like in 2021, the year that we built it, where like things haven't really changed yet. And then in 2025, 4 years later. Here's a few things I'm going to point out. You can see that there is a new zone of sediment deposition happening over there. This bank is also actively being eroded. The Thawag has shifted from kind of like this shape to southward like this, and the channel is much more defined. You can see it's not really dispersing and shallowing out like it looks like here. It's very clear where the water is and where the banks are. As you move downstream, there's a much more exaggerated bend in the stream channel. The channel is more defined in general, and all the material, eroded material from like this section here, has been deposited downstream onto this part of the bank here, and this little bar here. So that's super cool. New banks are being formed. This is when like erosion is necessary to be able to change the landscape, because you've got to take the soil material from somewhere to be able to put it somewhere else. So in this case, the erosion is good. It's what we're looking for here, because it's actively building new banks. Super cool. And here is, I've kind of like loosely drawn out the center line, the thou lake, like where most of the water is flowing in 2021 versus 2025. Here they are kind of superimposed on each other, so you can see just how much change has happened, how much shifted southward, all this like change in like the way that the creek is kind of like S-turning and meandering. Super cool stuff. I am not good at statistics. So we have been working with Mount Royal University to do some GIS, some analyses of all the data. We have all this orthoimagery data, but it just kind of sits there. I don't know what to do with it. I don't know how to read it, but they do, which is really great. So we're working with Dr. Lynn Mormon. Her 4th year GIS students are looking at this data, and every year they've been doing, just kind of working it up, coming up with their own questions and their own answers. So here's some of the preliminary data. This data has been vetted by Lynn as good data. So she's like, some of the stuff that the students have found is that there's a big increase in sinuosity. There's like a 2,600% increase in pools. And this stat is old too. This is from 2022 data. It's 2025 now. I'm sure that's changed. Here is a cool graph showing how the stream profile has changed. So this is a cross-section view. This is like essentially the bottom of the stream. And you can see that there's like new thing going up here and then a new pool forming here. There's been an increase in total stream length. So there's straight up more stream habitat, like 9 meters more stream habitat. And the riparian health, specifically shrub cover, is like way more robust and way more healthy. In addition to the GIS, we have also been doing annual electrofishing surveys. 300-meter reaches from like downstream is where we did most of the work. Upstream was our control reach, where we didn't do anything. There's still cattle impacts and everything there. And this is not statistically significant, or if it is, I don't know, because I haven't done any analysis. This is just straight up the numbers. So all I can say like with certainty is that there are now bull trout downstream, and there didn't used to be. Every year since we've done the work, we have found bull trout downstream. And to me, it appears as if there is some sort of flip, like there's more fish downstream than there are upstream where the impacts are still happening. However, not statistically significant. I don't know, I don't claim that. And since then, in 2024, we have built 20 more structures upstream. So in our like old control reach, so we can't really use that as a good control anymore, but we've done a lot more work there, because just because we're using it as a control doesn't mean we will let things like fade into obscurity. We want to help fish there as well. These 20 structures include not just post-assisted log structures, but also beaver dam analogs, which are, man-made analogs of beaver dams. And this is just kind of a call to everyone out here. If there's any students, academics, or people that think playing with R is a fun weekend activity, we have so much data here that we would love for someone to help us analyze and parse through. I don't know what a p-value is, so I will believe whatever you say to me. We have 5 years of orthoimagery data at Radiant Creek that you can work up, 2 years of data at Elk Creek, where we've also done this work, 2 years of data at Vipers Creek, where we've done a bunch of beaver dam analogs as well, and then upcoming data at Sharples Creek. We flew a drone flight back in 2024, and I think we want to do it again, in addition to some of the stuff that Ben's done with FRI, or the UFC, sorry. So yeah, lots of opportunities for student projects, for just studies in general, for if you think looking at this is a fun time. Happy to work with you, hand it over. I can't guarantee that it's great, but clearly it's usable according to some of the MRU students. And yeah, thank you to everyone that's helped us do it so far. We have funding from a few people, but like this work is super labor intensive, and it took a lot of volunteers to do it, including folks from Cows and Fish, Clearwater County, where this project is based in, as well as like we've had like 4 years of kids from the Junior Forest Rangers program come up and build beaver dam analogs and post-assisted log structures with us. And they always seem to have a lot of fun. And we still have lots of work to do there, so we always welcome your help and always recruiting volunteers. Thank you.
All right, we have just a couple minutes for questions. Do we have any questions right off the bat? Yeah.
I just want to say thank you, SCT, for taking on this project.
Thanks, Jess.
This is a great project, and I was there for 3 nights at work. I caught bull trout where no bull trout should be able to survive, so thank you. What do you think the future holds for raising creek? Like, is there an opportunity for long-term stream or what happen?
I think there is like, here, I'm just gonna go forward a slide. I prepared, in the upstream area, you can see there is like lots of wood and stuff that would normally be in the stream. It's just that the downstream area is so impacted. There's no potential for like any of that stuff to stay. It just kind of leaves because it's like so simple of a reach. So I feel like there is potential for more work to happen there. I feel like a lot of the impacts, Radiant Creek is easy in the sense that there isn't, the main impact there is grazing. So it is actually just one thing. Like we're not dealing with a bunch of OHV traffic. There's some camping, but it's not really happening adjacent to the stream and causing a lot of problems in that way. So I feel like there is a lot of opportunity for cool work to keep happening there, and we are very persistent and always keep pestering people to fix things. So I think that kind of helps move things along too.
Any other questions?
How did you restrict grazing on such a broad floodplain area?
A really big fence for this. In the project area, the 2020 project area, we have like a metal panel fence that I think we bought and we owned. Yeah, and it's just straight up installed all around. Every year we go in the spring and take it down so that when it floods, like before the floods, so it doesn't get buried. One year we didn't do it in time, and we lost a few panels that got buried in like 4 feet deep of sediment. And then we come back like in the fall, or we come back in the summer right before the cows go in to put the panels back in. So it is kind of labor intensive, I guess. And the fences are expensive, but I don't think the project would have succeeded if we hadn't excluded cows with all the fencing. So it was pretty crucial. No, there's no solar waters. There's no like water feed, like chirotraph trough or anything. It's just that like this is such a huge area, and we're not restricting 100% of the creek. We're restricting these patches where we're working, and there are places like little gaps in the fence. Like here is the upper part of Radiant Creek where we were working in 2024. You can see there's a little gap that we've left for cows to water, for the rancher to cross. So we're not fencing out 100% of everything, and we always, we talked to the allotment holder here, and that's why there's a gap there, because he was like, I need to cross here. So we were like, okay, we'll let you cross there. We're like, okay, you can cross there. We won't build anything there.
Excellent. All right, we are running a little short on time, so we'll leave it there for questions. Thank you so much, Angela. So, since the presentations have concluded, we are going to end the stream. So for everyone online, thank you so much for joining us. Thank you again to Mike, if you're still in the chat. Thank you so much for presenting again. Okay, so we are going to end the stream now. Can you end it on your side? We're getting claps for everyone presented. Okay.